The basic unit of eukaryotic chromatin is the nucleosome, a complex formed by a core of 8 histone proteins around which the DNA is wound for almost 2 turns. DNA that is complexed in the nucleosomes is not accessible by many proteins. To find out how nucleosome spacing varies along the chromosomes, we can use a method that uses DNAseI to digest chromatin, followed by separation of digested DNA fragments and sequencing. The method works like this. A nuclease such as DNAseI is incubated with native chromatin. The nuclease can access most easily stretches of DNA that are free of nucleosomes and thus are most exposed. Small DNA fragments are generated preferentially by digestion of these regions and can be separated easily from the larger, less accessible fragments. The fragments can be sequenced efficiently in sequencers such as those made by the Illumina company. Once the sequence of the small DNA bits is available, it is matched to the genome sequence of the organism from which they were isolated. The matches accumulate corresponding to the open DNA regions and not in genomic regions that were protected by nucleosomes. If we plot along the genome the number of matches, which we call read coverage, we obtain peaks that correspond to DNAseI hypersensitive regions, which we infer were devoid of nucleosomes. What is the biological significance of these sites? By comparing them to other genome annotations, such as known gene position and regulatory regions, we know that they fall in 2 main categories: promoters and enhancers. We conclude that the 200 to 300 base pair directly upstream of the transcription start site, which correspond to the promoter, need to be nucleosome-free to allow access of RNA polymerase and transcription factors to their DNA binding sites. The same applies to activators that need to access selected sequences in the enhancer regions. How do these regions become free of nucleosomes? Scientists believe that regions covered by nucleosomes can still be recognized by special prospector proteins. These pioneers have special enzymatic activities. They are nucleosome remodelers. They use the energy of ATP to push and shove nucleosomes, popping them off the DNA and freeing up certain specific regions. In conclusion, DNAseI hypersensitive analysis coupled to sequencing and genomic analysis has provided evidence for special DNA sites that are free of nucleosomes. These sites are found adjacent to transcription start sites and corresponding to enhancers. Special regulators act to keep these sites open to allow access of transcription factors and RNA polymerase, which can bind their target sites unhindered by the nucleosomes. We can access this analysis at websites called genome browsers. We're going to visit one called the Plant DHS database, where DHS stands for DNase hypersensitive site. Here is the genome browser for the rice genome. We are viewing chromosome 3, left to right, focusing on a small 23 kilobase window, indicated here. Let's turn on the gene track. This plots on the genome the information about genes. The genes are illustrated with exon boxes and intron lines. The orientation of transcription is symbolized by the arrow. Let's turn on the DNAseI hypersensitive track, which displays the analysis we described early in this video. The peaks that appear correspond to regions where DNAseI digested DNA was identified by sequencing. This peak is positioned between the 5' of 2 genes that are transcribed divergently. It corresponds to the promoter regions for both genes. Moving to the left, we find a small gene that has a hypersensitive site to the 3' only. Moving to the right, we see other genes. Some, but not all, have hypersensitive sites. Sometimes, hypersensitive sites are located away from the transcription start site, corresponding to additional regulatory regions. Returning to our original site and zooming in, let's compare the analysis carried out on chromatin from different tissues. On top is leaf data. which we were looking at. On the bottom is data from in vitro culture cells, a tissue called callus. We see that the same hypersensitive site is present, but a new one has also appeared away from any known genes. This is not a common finding. It may be an enhancer that is uniquely active in callus. We can turn on another related analysis that determined, using another method not described here, the occupancy, percent presence among all cells, and position site preference of nucleosomes. The important observation here is that hypersensitive regions are depleted of nucleosomes. Last, we can now compare these different analyses to another chromatin trait, histoneacetylation. The amino terminus of histone 3 has a lysine at position 9, abbreviated as K9, whose acetylation can be measured accurately through yet another method not described here. Histoneacetylation is typically associated with well-expressed regions. Turning on that track, we find that the histoneacetylation signal from the hypersensitive site is very strong compared to the rest of the region. Their position indicates that the nucleosomes at the beginning of each gene are strongly acetylated, perhaps making them easier to move by chromatin remodeling proteins and the transcription apparatus. To summarize all this, let's look at another example. Here is a gene transcribed left to right with a strong hypersensitive region at its 5', a nucleosome depleted region, and a decent H3K9 acetylation signal immediately downstream of the hypersensitive site. The cartoon illustrates the corresponding regulatory features.